the location of its geographic range boundary. So we have a species occupying some area. We might think that given enough time, the peripheral population of that species can go through successive bouts of local adaptation and grow the range size kind of like a tree accumulates rings, to paraphrase Ernst Mayr. But what we see in nature is that all species have restricted distributions, so they vary a lot in size and shape. And barring dispersal limitation, ultimately this is an evolutionary problem. So the species occurring in green here is simply not adapted to the environment outside its range. Approximately, this is an ecological issue, where it's the interaction of the organism with some aspect of the novel environment outside its range that is keeping fitness below population sustaining levels. And we usually think about this theoretically as a one-dimensional environmental gradient shifting across space. But as we all know, the environment is multidimensional. You can see from this rainbow laser beam here. And multiple environmental variables often co-vary across space. And the fitness of any one population distributed across this gradient is really determined by the matching of the population trait value to the optimum trait value dictated by the environment. So as the environment changes across space, our phenotypic optimum changes, and the degree of mismatch between that phenotypic optimum and the actual realized trait value of the population determines the degree of maladaptation. As those points diverge, we can get this hard range limit. And a while ago, Antonovics told us that given the multivariate nature of the environment, we might expect that for any species to invade a novel range, we should expect that multiple traits might have to evolve simultaneously to deal with multiple aspects of the environment. But to throw another wrench in the machine here, the environment, these gradients themselves can actually directly influence phenotypic expression in species. And if that phenotypic plasticity is in a direction antagonistic to the direction of selection across space, that can further depress fitness in already marginal environments. So, just kind of general concept, spoiler here, I'm going to be talking about a suite of abiotic and biotic factors that vary across space, across the species range. Talk a little bit about the traits that mediate responses of organisms to these clines, and then talk about how the actual clines themselves feed back into phenotypic expression of the traits. So I work with Clarkesantiana, which is a narrow endemic wildflower in the southern foothills of the Sierra Nevadas. And it's comprised of two subspecies, Clarkesantiana in the west and Clarkesantiana parviflora in the east. And there's good population genetic evidence that these two subspecies have gone through a classic case of ecological divergence in allopatry and are now in secondary contact at a narrow range of parapatry right around Lake Isabella. And we're going to, when we plant these subspecies into each other's ranges, we see fitness decline markedly. And we're going to focus today on the western Xantiana and its eastern range limit, which is this red line here. And when we move Xantiana over this red line into parviflora territory, we see fitness decline by about 90%. And so what this sets up is a situation where we have two taxa that are very closely related. They occupy regions directly adjacent to each other, but they show strong adaptation within their home ranges and strong maladaptation between their ranges. So what we can do is we can look at how the environment is varying across this space, kind of map that on to phenotypic differentiation between the subspecies to get an idea of kind of the factors that might underlie this hard range limit. And so a lot of cool work that others have done before me has identified a lot of this. So with the environmental variables, we see aridity increasing as we move east, and we see pollinator communities getting less diverse and less abundant. We can kind of map this on to phenotypic differentiation between the subspecies, where we see delayed phenology in the western subspecies where the environment is relatively wetter, and then in the arid east we see this very rapid development that we usually associate with arid land plants kind of escaping late season drought. The pollinator cline seems to map up with this difference in mating system where we have a predominantly outcrossing subspecies in the west, Xantiana, and predominantly selfing subspecies in the east, of course. But as I said, this is a complex environmental gradient, and when they've planted Xantiana across this red line into parviflora territory before, they've seen that just as many plants seem to die from fatal mammalian herbivory as do from water stress. So I wanted to get a finer scale mapping of this potential cline and herbivory. So across 14 sites from the center of Xanthena's range out into parviflora territory, I did a translocation experiment. So one nice thing about Xanthena, other than being beautiful, is that you can cut the stems, put them in water, and the plant continues to develop, it opens flowers and sets seeds. And so what this means is we can move plants anywhere we want to in the range without having actual rooted plants. So you can see what the plants look like in the field. I made these little tubes out of 50 mil falcon tubes, camouflage the tops. We can set these into the ground and put stems in them and look at herbivory across the range. So left them out for 5 days, and then you can see some of the herbivory that we see, which is this clipping right at the base of the stem. And what we see is, here we're looking at the percent fatal herbivory by site, plotted against easting. So the center of Xanthena's range is on the left side, x-axis. This is the range limit, and then we're moving outside the range. And we see that at the center of Xanthena's range, we have fairly low rates of herbivory. But right at the range edge, that starts going up, and we can get really extreme values just outside the range. And this is best fit by an exponential function. And we're also able to identify some of the perpetrators here. This is the black-tailed jackrabbit, Lepus californicus, but we also see things like deer and small rodents attacking the plants. So we can kind of decompose this multivariate environment into a few key aspects. We see herbivory and aridity increasing as we move east, and pollinator communities getting less abundant and diverse. And focusing on herbivory, then we can ask, all right, are there traits that underlie adaptation to this environmental climate in herbivory? And so to look at this, I analyzed data from the first transplant that Monica Gaber and Vince Eckert did, where they took both subspecies and planted them into 3 sites, one at range center, one at the edge of Xanthena's range, and one outside the range in parviflora territory. And what I'm showing you here is the relationship between probability of herbivory, these are predicted probabilities from logistic regressions, plotted against phenology on the x-axis. And what we see that's really cool is this relationship changes across space. So there's no relationship between phenology and your probability of getting eaten at range center, but at the range edge, and especially when you go outside the range, we have this very strong relationship between phenology and herbivory. And you can see the 2 subspecies sorting out over phenological space here, where we have parviflora in the white circles. They're all clustering on the left side of the x-axis, so they develop very fast and escape herbivory. Xanthena in purple circles is on the right, develops very slowly, and is very susceptible to herbivory. And if we analyze Xanthena by itself, with just purple circles, you see that outside the range, every day of delayed flowering increases the odds of herbivory by about 17%. So there's a very strong relationship between a specific trait and this environmental climate. We kind of fill in some of these gaps here in the concept map. But as I said, the environment itself can actually have this effect on phenotypic expression. And an interesting thing that we see in this transplant experiment is if we look at the distribution of phenology just for Xanthena across these 2 sites, even though selection would favor very rapid phenology in the east, at this eastern transplant site, we actually see the distribution of phenology shifted later at that eastern site outside the range than phenology at the center of the range. And so this is probably driven by a few different things, but since the word microbes is in the title of this talk, that's the one I'm going to focus on today. And I was interested in, given the strong environmental climes across this range, we're increasingly seeing that both small and large-scale spatial variation in microbial communities can drive phenotypic variation in plants. So I wanted to look at this with Xanthena. So in the greenhouse, I grew Xanthena plants with either soil microbes sourced from the same site as the seeds are sourced, or foreign microbes from other Xanthena sites or other parviflora sites. And given the strong gradient in aridity across this climb, I crossed this in a full factorial design with a high and low watering treatment and stored these for growth and phenology. And what we see is when plants are with foreign microbes, their phenology is delayed by about 2 weeks. But there's no difference in reproductive effort. So it's not that with local microbes you just do really poorly and you develop quickly. You produce the same number of flowers, but your whole life cycle finishes 2 weeks faster. So here we're just plotting local at the top, foreign at the bottom. but this varies by watering treatment. So it's only under water stress do we see this effect of microbial communities. And so what this tells us potentially is that if Xanthia is invading a novel region, if seeds are going outside the range limit, but microbial communities are not shifting alongside that, we could have this potentially important shift in phenology for the species. And to map this onto the relationship I showed you earlier between phenology and herbivory, 2 weeks doesn't sound like a very long time, but this, given the strong relationship here, this could take a plant from about 30% probability of herbivory to 60% probability of herbivory. So this potentially be a very important environmental driver of this trait. So what I've shown is there are a lot of things changing across this gradient as we move from the range limit of Xanthina into parviflora. Kind of focused on this biotic interaction of fatal mammalian herbivory and how that changes as we go toward and beyond the range limit. And we're able to see that susceptibility to that herbivory is largely driven by a single trait, phenology, but that relationship varies across space, so only at the range edge and outside we really see this relationship. But that trait itself is actually influenced by environmental variation across space, so expression of that phenological phenotype. And potentially part of the story here is that microbial communities shifting across this range can drive some variation in phenology. So we have this complex interplay of these different abiotic and biotic variables. So we can identify all these different pieces or these factors influencing range limits, but what we'd really like to do is estimate some sort of effect size, right? So if we can get at what's the relative influence of all these different factors. And the way you can do this is through the old kind of kick it and see approach, so manipulative transplant experiments, where we can protect plants from herbivores. I'm doing some now where I'm actually moving microbial communities around in the field and manipulating precipitation. For the pollinator clines, we can supplement pollen to kind of overcome pollen limitation. And then we can actually feed the data from those experiments into population projection models. And we can ask, all right, well, if we protect plants from herbivory, what does population growth look like outside the range for a new population establishing there? Or if microbial communities were shifting along with the plants, would that enable range expansion? And what this gives us is a more mechanistic understanding of the ecological factors, kind of structuring range limits. And then if you pair this with a lot of cool work that my advisor and his collaborators are doing, looking at more of the kind of evolutionary and genetic forces that may constrain adaptive evolution in these important traits at range margins, we can start to get some good empirical evidence about what may be limiting adaptive evolution at range margins and structuring species distributions in the wild. So with that, I'll thank folks that gave me a bunch of help and funding, and I might have time for questions. I'm not expecting that.